Okay, so here we are in the skin cell again, and I want to show you why certain genes are turned off and certain genes are turned on. And so DNA is wrapped around, so this is the DNA here, this blue ribbon. It's wrapped around these complexes of 8 proteins called histones. So each one of these is a histone, and the histone has the amine terminus of the protein kind of sticking out in a long tail. So there's a tail for every histone protein. And the group of all 8 histones is called the nucleosome, and the nucleosome has the DNA wrapped around twice. And so when you have a bunch of nucleosomes, they're actually called beads on a string. You just have a lot of these one after the other, and it helps the DNA to be compacted, just like how a spool is helpful to organize thread. The nucleosomes are useful to organize the DNA. But there's also these little chemical groups that are on the histone tails that can either be positive or negatively charged, and depending upon their charge will depend on whether the DNA is going to be tightly wound or whether it's going to be loose. And when the DNA is loose, then RNA polymerase actually has a chance to be able to get in and transcribe genes, so the genes can be on. But when the DNA is tightly wound, there's no way for RNA polymerase to get in there. So right now the way I have it is these little beads are representing negative charges on the histone tails. When those are present, positive charges on the histone tails. When they're present, these positive charges attract to the negatively charged DNA, and it brings the histones close to the DNA. So like these will be attracted to this DNA, and these will be attracted to that DNA, and it's just going to bring these close together. So then the genes will be off because RNA polymerase won't be able to get in there. So to turn the genes on, you need to loosen the chromatin. And an enzyme does that. And what it does is it adds these acetyl groups. So the acetyl groups are a way to modify the histone tails. So the negative charges are removed, and these acetyl groups are added. And the acetyl groups mask the charges, so these will no longer be positively charged, and so they'll no longer be attractive to the DNA, and so that will cause the nucleosomes to be further apart. The DNA can also be modified to turn genes on and off. In general, methyl groups, which are CH3 groups, if they're added to the cytosines or guanines of the DNA, that attracts an enzyme called histone deacetylase, which will remove the acetyl groups, and the negative charges will return. So if you want to turn a gene off, a lot of times the DNA is methylated, which causes the acetyl groups to be removed, and then you restore the positive charges, and then the DNA can go together. So methyl groups turn genes off because they wind up the DNA more, and acetyl groups on the histone tails turn genes on.